What is the response our body and mind has on a stimulus? How does our body respond? It's neurons and neuronal communication. In our nervous system, there are thousands, millions, and billions of neurons that help with the communications within our nervous system. So what does a neuron look like? What is the anatomy of a neuron? In this case, we'll take a closer look at a multipolar neuron. The cell body, or soma, is a metabolic center. The cell membrane is what encloses the neuron, or is the wall of the neuron. The dendrites are tree-like branches that receive most synaptic contracts or signals from other neurons. And the axon hillox is a cone-shaped region that connects the axon and the cell body. Taking a look at the other half of the neuron, the axon is that long, narrow process that projects from the cell body. The myelin sheath is a fatty insulation around the axon that allows for electrical impulses to travel quickly and much more efficiently. The nodes of Ranvier are the gaps between the sections of the myelin, and the terminal buttons on the end of the axon branches are where chemicals are released into the synapse, which I'll go over in a bit. So these are the main parts of a neuron, and a little bit about its function. When we zoom in and take a closer look into a neuron, we can see this layer called, which I mentioned, the cell membrane. The cell membrane is a lipid bilayer with signal proteins and channel proteins embedded in. We'll now get into how neurons work and how they communicate. Looking at the cell membrane, the membrane potential tells us the positively or negatively charges inside and outside the cell. These charged particles, which are called ions. When a neuron is steady, meaning no action is taking place, or the resting period, is when a neuron is polarized, or it's at its resting potential of about -70 millivolts. During this resting period, focusing on just two ions, potassium and sodium, there are more potassium ions on the inside and more sodium ions on the outside. The distribution of these ions are done so by ion channels that allow only certain ions to pass. During this resting phase, only the potassium channels are open, but only a few potassium will exit out. Going back to the neuron, when neurons fire, the terminal buttons release chemicals called neurotransmitters that diffuse across the synapse. The synapse is that gap between the terminal buttons and another receiving neuron. When the neurotransmitters are released, they bind to postsynaptic receptors on the membranes of the next receiving neuron. Here, one out of two things occur: depolarization and hyperpolarization. When depolarized, the resting membrane potential decreases. This is also called the excitatory postsynaptic potentials, since the likelihood of neurons firing will increase. When hyperpolarized, the resting membrane potential increases. This is also called the inhibitory postsynaptic potentials, since the likelihood of neurons firing will decrease. So these EPSP and IPSP signals then travel down at rapid and decremental speed. The firing of a neuron, depending on the EPSPs and IPSP signals, generated in the axon's initial segment, located right before the axon here. This firing is an action potential, which lasts for 1 millisecond and is an all-or-nothing response. An action potential occurs when it reaches the threshold of excitation, which is -65 millivolts, when the membrane is depolarized. It will then use individual signals from EPSPs and or IPSP postsynaptic potentials to then tell the neuron whether to fire or not, and this adding is called integration. In an action potential, there are three phases. The first phase is the rising phase. So when the membrane potential is depolarized, the voltage-activated sodium channels open with sodium atoms coming in. Then the voltage-activated potassium ion channels open with potassium ions exiting outside the neuron. In the second phase, repolarization, 1 millisecond after the other phase, is when the sodium channels close. In the third final phase, hypopolarization, is when the potassium channels start to close. From an electrical to a chemical step, at the synapse, neurotransmitter molecules are then released and they bind to the receptors on the postsynaptic membrane. The presynaptic membrane is part of the sending cell, and the postsynaptic membrane is from the receiving neuron, which is on the other neuron. So the release of neurotransmitters is a process called exocytosis. So while at resting state, the synaptic vesicles will store these small molecule neurotransmitters and will then congregate near the presynaptic membrane, ready to release the neurotransmitter. So once an action potential occurs, the channels will then open and calcium ions will enter the terminal button. But because of the calcium ions entering, this will cause the synaptic vessels to merge onto the presynaptic membrane, and from there it will release the neurotransmitters into the synaptic cleft or synapse. After the neurotransmitters have sent their message, there are two types of mechanisms that occur to remove the remaining neurotransmitters from the synaptic cleft. So 1. In reuptake, neurotransmitters are drawn back to the presynaptic buttons by transporter mechanisms. In enzymatic degradation, enzymes, which are proteins that brings about a biochemical reaction, breaks down neurotransmitters. Afterward, the parts are recycled up through the transporters to be used again. This is the end of what a neuron is, its functions, and how communication signals pass through the neurons, both electrical and chemical. Thank you for listening.